Have you ever wondered how the food you eat becomes energy inside your body? Today, we will discuss glycolysis, that is the first and most important step of cellular energy production, and how glucose enters the cell with the help of insulin and glucose transporters. After digestion, glucose enters the blood, but glucose cannot enter cells by itself. For muscle and fat cells, insulin acts like a key. Insulin binds to its receptor and opens special doors called GLUT4 or glucose transporters, allowing glucose to enter the cell. In brain cells, glucose enters without insulin using GLUT1 and GLUT3 transporter because the brain always needs energy. Once inside, glucose is ready for glycolysis. Glycolysis happens in cytoplasm of the cell. It breaks one glucose molecule into two pyruvate molecules. This process has 10 enzymatic control steps and produces two ATPs, two NADH, and two pyruvate. The key regulatory enzymes include hexokinase, phosphofructokinase 1, and pyruvate kinase. Now we will discuss the glycolysis stepwise. So after entry in the cytoplasm, glucose is first phosphorylated into glucose 6-phosphate by the enzyme hexokinase or glucokinase, utilizing one ATP molecule. The glucose 6-phosphate is further converted into fructose 6-phosphate by the enzyme phosphoglucose isomerase. The fructose 6-phosphate is then again phosphorylated at carbon number 1 to fructose 1,6-biphosphate by the enzyme phosphofructokinase 1 utilizing another ATP. The fructose 1,6-biphosphate is cleaved by the enzyme aldolase into two products, that is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. The dihydroxyacetone phosphate is further converted into glyceraldehyde 3-phosphate by the triose phosphate isomerase enzyme. The glyceraldehyde 3-phosphate is converted into 1,3-biphosphoglycerate by the enzyme glyceraldehyde 3-phosphate dehydrogenase, which remove hydrogen from the glyceraldehyde 3-phosphate and add it into the NAD, converting NAD into the NADH. 1,3-biphosphoglycerate is further converted into 3-phosphoglycerate by the enzyme phosphoglycerate kinase, which convert one ADP molecule to ATP molecule. The 3-phosphoglycerate is converted into 2-phosphoglycerate by the enzyme phosphoglyceryl mutase enzyme. The 2-phosphoglycerate is then converted into phosphoenol pyruvate by the enzyme enolase, releasing one water molecule and converting the 2-phosphoglycerate into phosphoenol pyruvate. In the final step, the phosphoenol pyruvate is acted upon by an enzyme that is known as pyruvate kinase, which convert phosphoenol pyruvate into pyruvate, and the released phosphate group is added to the ADP, which is converted into ATP. Thus, one glucose molecule is converted into two pyruvate molecules.